Welcome back to this next video in which we are discussing the control of blood pressure and blood volume. Okay, so we're currently in the process of discussing the Renin-Angiotensin-Aldosterone System, which is a system that is going to be activated if mean arterial pressure goes too low, and is going to be inhibited if mean arterial pressure goes too high. And we've talked about the ways in which it's going to help to renormalize blood pressure by altering the constricted state of blood vessels all over the body, and also by altering blood volume. Okay, so far what we have seen is the cells that are actually involved in releasing renin, the first component of the renin-angiotensin-aldosterone system, and these are of course these granular cells here in blue. And we've seen how a decrease in mean arterial pressure is going to lead to increased renin release above the basal level, whilst an increase in mean arterial pressure is going to lead to a decrease in renin release below the normal basal level. Okay, so now what we want to see is what is renin actually going to do. So I've discussed that renin is an enzyme, and it's actually going to break down a protein that is in the blood that is put into the blood by the liver. And this protein is known as angiotensinogen, which means the precursor to angiotensin. So it's going to break down a protein known as angiotensinogen, which is put into the blood by the liver. And just to draw a little picture of this, angiotensinogen is a very large protein that is circulating within the bloodstream. So here it is, drawn very simply. Of course, in reality it will be folded up into a tertiary protein structure. However, just for simplicity, I'm drawing it now just as a linear polypeptide like so. So here is my picture of angiotensinogen. It's circulating in the blood. It doesn't do anything before the renin has actually been released into the blood to break it down, but it is put into the blood by the liver, and it is a very large protein that is circulating within the blood. So, what's its role then now in the renin-angiotensin-aldosterone system? So as I say, before it's been broken down by renin, it doesn't do anything. It's just the precursor to angiotensin. So the renin that has been released into the blood by these juxtaglomerular cells or granular cells is going to break down angiotensinogen. It's going to cleave one of the peptide links in the angiotensinogen polypeptide. Specifically, it's going to cleave the link between the 10th and 11th amino acid. So if you count your way along the amino acids from the amino terminus to the carboxy terminus of angiotensinogen, it'll take you a long time. There's a lot of amino acids in here, but we're not going to go far. We're just going to go to the 10th amino acid, and specifically we're going to look at the peptide link between the 10th and the 11th. So let's say here is the 10th amino acid, and then the next one along is the 11th amino acid. We are going to cleave that link between the 10th and 11th amino acid of angiotensinogen, and overall the result of that then is that you'll now end up with two fragments, one which is from the amino terminus to the 10th amino acid, and then the other which is from the 11th amino acid onwards to the C-terminal amino acid. We care about this first amino terminal fragment, the fragment of 10 amino acids here. Okay, so this cleavage is catalyzed then by the renin enzyme. So the renin that is released into the blood is now going to work on angiotensinogen molecules that are in the blood and cleave them in this way between the 10th and the 11th amino acid to produce this 10 amino acid little fragment here. And this 10 amino acid little fragment here is now known as angiotensin 1. And for short, angiotensin 1 is often abbreviated down to Ang for angiotensin, and then you just put 1 after it like so. And often you use Roman numerals rather than the more normal numbers, so you can put an I like that for 1 instead of putting the number 1, but either would be understood. Okay, so this is angiotensin 1, this decapeptide of 10 amino acids. And by the way, when you put alpha alpha like that, just in case anyone doesn't know, that means amino acid. Okay, so we can describe angiotensin 1 then as a decapeptide. Now, angiotensin 1 isn't particularly active at all, so it has to now be activated further But once you've produced angiotensin 1 because of the renin, angiotensin 1 will be converted to the active form without any further enzyme having to be released into the bloodstream. So angiotensin 1 is now going to be converted to the activated form, which is angiotensin 2. And this is the one that's going to have lots of effects. So angiotensin 2, and again, usually we use Roman numerals rather than the number in this case. So you wouldn't usually see people actually write 2 here, you'd see them writing the Roman numeral. And again, the shorthand then for angiotensin 2 is Ang2, like so. Okay, so angiotensin 1, once it's been produced by the renin, is going to be converted to angiotensin 2, and the way in which this is done is you just chop another 2 amino acids off. So angiotensin 2 is an octapeptide. It's going to have 8 amino acids, and you're going to chop the 2 amino acids off from the C-terminal end of angiotensin 1. So you've got 10 amino acids here. Go to the end and chop off the 9th and 10th amino acid. Cut the peptide link between the 8th and the 9th amino acid, and you then get a fragment of 8 amino acids and a dipeptide of 2 amino acids. The fragment of 8 amino acids is the one that we're interested in. That's our angiotensin 2, and this is the active molecule now. This is actually going to biologically do something. So how does this conversion occur? Does it just occur spontaneously? No, there is an enzyme that is needed to catalyze it, but this enzyme is always available unless you're taking some sort of medication that inhibits it. And this is the ACE enzyme, the target of the drugs, the very famous antihypertensive drugs, the ACE inhibitors. So ACE stands for angiotensin converting enzyme. So it's a sensible name. It's going to convert angiotensin 1 into angiotensin 2. Okay, now where is the angiotensin converting enzyme found then? Is it within the bloodstream? Well, no. It's on the surface of endothelial cells all over the vasculature, but particularly in the pulmonary vasculature is the classical site that people say the angiotensin converting enzyme is present. So what do I mean by on the surface of the endothelial cells? Well, if I just draw a little picture of an endothelial cell here. So here is an endothelial cell. Endothelial cells, they do not have a simple apical membrane, okay? It's not just an array of phospholipids. If you were a little man standing here looking down at an endothelial cell, you wouldn't just see an array of phospholipids. Instead, what you'd see is attached onto the apical surface is a meshwork of molecules, proteins and carbohydrates all over the surface of the endothelial cell. So the membrane would be littered with proteins, integral membrane proteins. You'd have them all over the place. So here might be a few integral membrane proteins. And then attached to the integral membrane proteins, you'd have loads of fibers, carbohydrate fibers in between them. Okay, so here are carbohydrate fibers, and then attached onto those carbohydrate fibers and also protein fibers you would also have. So you've got this meshwork, a spider's meshwork on top of the phospholipid bilayer, and attached onto that you can then have enzymes sitting on. So it's a very complicated thing, the apical surface of an endothelial cell, and this is where the angiotensin converting enzyme can be found. That's what I mean by it can be found on the apical surface of endothelial cells, attached to this carbohydrate fiber meshwork that is on the apical surface of an endothelial cell. And this meshwork that's on the surface of an endothelial cell, this has a special name. It's known as the glycocalyx, like so. Okay, so the ACE enzymes then can be attached to the glycocalyx of endothelial cells. And as I say, you find ACE enzymes on the surface of endothelial cells all over the body. So go anywhere in the vasculature in the body, you'll find angiotensin converting enzymes. But particularly the famous site where you can find angiotensin converting enzymes is if you go into the pulmonary vasculature, so the pulmonary circulation. Okay, that's where you can in particular find angiotensin converting enzymes. But the point is that they are readily available to convert angiotensin 1, which is in the blood, into angiotensin 2, which is in the blood. So the instant you produce angiotensin 1, it will be converted to angiotensin 2 by ACE enzymes. So this is not a limiting control point for the production of angiotensin 2. The control point is the conversion of angiotensinogen into angiotensin 1. Okay, right. So, produce renin, it's going to result in angiotensin 2, and this is the lovely bit. This is the arrow, single dimension flow diagram here, so very, very simple. So, renin release from the granular cells, overall, that results now in the production of angiotensin 2 molecules in the bloodstream. So, if mean arterial pressure is too low, angiotensin 2 is going to go up. If mean arterial pressure is too high, angiotensin 2 is going to go down. Very simple now. Now what I want to see is what are the effects of angiotensin 2, and I'm going to talk about two of the effects of angiotensin 2. There are lots of them that you can go off and learn a list of if you like. I'm just going to dwell on the two main ones. So the first one is, why is it called angiotensin? Tense, okay? It's going to cause contraction of some sort of muscle cells. Angio, it's going to tense the blood vessels, so it's actually going to cause vasoconstriction. So this is the molecule then that is actually capable of vasoconstricting the vasculature all over the body. Okay, so the arteries, the veins of the arterial and venous system, the arterioles, all of them alike are going to be constricted by angiotensin 2. Now, how does angiotensin 2 actually work? Well, it's going to go to the vascular smooth muscle cells. It will leach out of the blood to the vascular smooth muscle cells of the tunica media of your blood vessels, and it's going to work on a receptor on these vascular smooth muscle cells, which is a G protein coupled receptor by the name of the AT1 receptor. Now, people always think that interpret the name of this wrongly. They say angiotensin 1 receptor. No, it is not a receptor for angiotensin 1. This is the angiotensin. The A is for angiotensin. The T then is for 2, so angiotensin 2 receptor subtype 1. So there are different subtypes of receptors for angiotensin. This is the subtype 1 receptor for angiotensin 2. Okay, so do not interpret that as a receptor for angiotensin 1. It is not. It's a receptor for angiotensin 2. So the AT1 receptor, the angiotensin 2 receptor subtype 1. And this receptor is going to couple to the GQ cascade, which will overall result in the release of calcium from the endoplasmic reticulum inside these cells into the cytoplasm, and that calcium will then drive the contraction of the vascular smooth muscle cells. So the higher is the activation of the AT1 receptors, the higher is the contraction of these vascular smooth muscle cells, and therefore the greater is the constriction of blood vessels. So angiotensin 2 causes contraction of vascular smooth muscle cells. What else to say? Okay, so this is going to occur all over the vasculature. So it's going to occur, as I talked about before, in the venous system, which is particularly important. So this contraction going up in the, let's say, venous system, what's the importance there? Well, of course, that will raise mean venous pressure, okay? And raising mean venous pressure will then lead to cardiac output going up in the way that we have seen before. This is the reason we had to look at that more complicated model of the cardiovascular system to understand why there is that interrelation. Okay, so mean venous pressure going up, we understand that that will increase cardiac output, and that will help to bring obviously mean arterial pressure back up. In addition, you're going to get contraction of the arterioles, and that will obviously raise total peripheral resistance, which will also help to bring blood pressure up. You'll also actually get direct contraction of the vascular smooth muscle cells around the arteries, and I'm running out of paper, damn it. So arteries here. Okay, and of course that's going to directly help to bring blood pressure up. So all of these ways then are going to help to bring blood pressure up as a result of contracting vascular smooth muscle cells all over the body. Effectively, we're just reducing the size of the vascular system. We're just constricting everything, and you can imagine that that's going to bring blood pressure up, the plain common sense there. Okay, so that's what's going to happen then if mean arterial pressure goes too low. Angiotensin 2 is going to go up. We're also going to see that angiotensin 2 will do other things apart from this, but I just want to summarize this effect of angiotensin 2 for mean arterial pressure going down and mean arterial pressure going up. So to summarize then this bit, if mean arterial pressure goes down, angiotensin 2 will go up in the bloodstream, cutting all of this bit out here. that will then cause constriction of all of the blood vessels around the body by activating angiotensin II subtype 1 receptors on the surface of those vascular smooth muscle cells. So you'll get vasoconstriction of the venous system blood vessels, which will cause mean venous pressure to go up, and therefore cardiac output will go up because we're pushing more blood now into the heart each time, and we know that by Starling's law that's actually going to result in an increase in stroke volume. We're also going to be increasing total peripheral resistance because we will constrict some of our arterioles, and that will make it more difficult for blood to return from the arterial system to the venous system, so that will help to bring up mean arterial pressure. And we're also actually going to be contracting our arteries, and that will obviously increase arterial blood pressure. So all of those things are going to help bring mean arterial pressure up. In contrast, if mean arterial pressure goes down, if mean arterial pressure goes too high, then the response of the renin-angiotensin-aldosterone system is to decrease angiotensin II from the basal level. So there is some angiotensin II always in the bloodstream. There is some activation of the renin-angiotensin-aldosterone system always. We haven't seen aldosterone yet. At the moment, we're just seeing the direct effect of angiotensin II. We'll come on to aldosterone. That's all about blood volume changes. So angiotensin II will go below the normal rate. That will then lead to less activation of AT1 receptors all over the vasculature, and therefore you'll get relaxation of vascular smooth muscle cells all over the vasculature. In the venous system, this will lower mean venous pressure and therefore lower cardiac output. It'll also lower total peripheral resistance by relaxing some of the vascular smooth muscle cells around arterioles, which will then vasodilatate, and we're also going to be relaxing the actual arteries, and therefore that will lower mean arterial pressure directly. So that's then the response to mean arterial pressure being too high. So now that we've done that, let's now look at the other effect of angiotensin II, and we will spend the rest of the video talking about the other effect of angiotensin II. So the other effect of angiotensin II is to activate the release of another hormone. So angiotensin II is going to bring about an increase in blood volume, and it's not going to do this directly, however. It's going to activate the release of another hormone called aldosterone, which is then going to increase blood volume. So for our discussion, we'll go back to the case where mean arterial pressure is too low, we're imagining, and angiotensin II levels have gone up, and then at the end we'll talk about what would happen if mean arterial pressure was too high. Okay, so angiotensin II is high in the bloodstream. The other great effect of it is that it's going to drive the release of aldosterone by a layer of the adrenal glands. So angiotensin II is going to go down to the adrenal glands, which sit atop the kidney, and I'll just draw a picture of an adrenal gland. So here then is roughly what an adrenal gland looks like. So the kidney would sort of be sitting under here, much bigger than the adrenal gland. So I'll just label this up. So this is an adrenal gland, and if you really want to be fancy, you can call them suprarenal glands, but I've never actually heard anyone except in anatomy class referring to these as suprarenal glands. Everyone calls them the adrenal glands. Oh, oops, and I've just written out anatomy. Suprarenal gland is what I meant to write out. Okay, right, it's been a long day. So, adrenal, suprarenal, whatever you want to call it, this is the gland that we're now talking about. And the adrenal glands can be split up into two layers. There is the adrenal cortex again, and then there is the adrenal medulla. So the same old Latin words. And there also will be a capsule around the edge of the adrenal gland, but we won't mention that. Okay, so we'll then have the adrenal medulla here in the middle, and then we've got the adrenal cortex around the outside. So I'll label this up. This is the adrenal cortex around the outside, all of this portion here, and then we've got the adrenal medulla in the middle. So this time we're going to start off in the center and we're going to work outwards. So the adrenal medulla, this contains the adrenal chromatin cells, which are the cells which actually release the hormone after, well, the central hormone of the adrenal glands, which is adrenaline. So the adrenal medulla contains the cells which release adrenaline. So the major hormone that this is going to release is adrenaline. This one isn't the one that we're actually interested in. Now adrenaline, of course, also has another name in the States, which is epinephrine. So if you're from the States, you'll probably prefer that name. we're not interested in this. We're going to be interested in aldosterone, and aldosterone is not released by the cells of the adrenal medulla. Now, let's go back then to the adrenal cortex. So the adrenal cortex can be split into 3 separate layers. And I'm now going to try and do this. So I'll colour the 3 separate layers in a different colour. So there is the most middle layer, okay, the most visceral layer, the deepest layer, which is a layer that synthesizes sex steroids, so sex hormones. And this is known as the Zona Reticularis. So this layer in red here is called the Zona Reticularis. And I should say that all of the layers of the adrenal cortex, they synthesize steroid hormones. Adrenaline is not a steroid, so the medulla doesn't synthesize steroid hormones, and I'll talk about what a steroid is for those who aren't completely clear on that concept in a moment. So Zona Reticularis is this innermost layer, and this synthesizes sex hormones, so testosterone, other androgens, and then the female sex hormones as well in women, of course. Okay, so that is Zona Reticularis. Then, around Zona Reticularis, you'll then have Zona Fasciculata, which is again a layer that we are not particularly interested in. This layer predominantly secretes the steroid hormone cortisol, which is the one that is released all the time to an extent. Its release is varying on a circadian basis. But it's also released a lot at times of stress. So this is the Zona Fasciculata here in blue. And then the final layer on the most peripheral layer is the layer that we're actually going to be interested in. So this is the Zona Fasciculata. I'll just underline that then in blue. And then finally, the outermost layer of the adrenal cortex, which I'll now colour in orange here, all of the remaining portion, this is called the Zona Glomerulosa. So it's got a name that's nicely easy to remember, that it's associated with the kidneys. And this is the layer that's going to release the steroid hormone aldosterone. So in orange there, this is Zona Glomerulosa. And the hormone that this is going to be releasing is, as I say, aldosterone, and the cells of Zona Glomerulosa are going to be activated to release aldosterone by angiotensin 2. So, firstly what I want to do is actually discuss what a steroid hormone is, then I want to show you the actual structure of aldosterone, and then we'll talk about angiotensin 2 causing the release of aldosterone by the Zona Glomerulosa, and then of course we'll go on to what aldosterone is going to do to actually raise blood volume. Okay, so firstly let me just talk about what is actually meant by a steroid hormone. So this is something that people often find surprising when they learn it for the first time. A steroid hormone is not defined by its biological activity at all. It's defined by a chemical structure. So when I, long ago, before I had been educated in medicine, I would have guessed that a steroid, just from popular understanding of the word, was defined by its biological activity. It was some hormone that had incredible effect on the body. I would not have thought that its definition was just a chemical structure definition, but in fact steroid molecules are defined based on their chemical structure. So you decide whether a molecule can have this term steroid applied to it based on its chemical structure. And a steroid molecule has to contain a basic core structure, the steroid core structure. And this steroid core structure consists of 17 carbon atoms with lots of hydrogen atoms coming off them that form 4 connected rings. So what I'm going to do is I'm going to draw the skeletal structure of a steroid core nucleus. And this is going to look very simple because remember in skeletal formulae we don't show carbon atoms, we show them implicitly by the corners of the diagram, and we don't show hydrogen atoms coming off carbon atoms. They're implicitly shown by carbon atoms having missing bonds coming off them. And of course the steroid core structure, it's just going to consist of carbon atoms linked to other carbon atoms with hydrogens coming off. Hence it's going to appear very, very simple when we draw its skeletal formula. So let me draw this then. So it's going to consist overall of 4 rings. I'll draw these like so. So here's the first ring. Here then is the second ring. Here is the third ring. So 3 6-membered rings so far. And here then is the 4th ring, like so, which is the only 5-membered ring. So this is the skeletal structure of the steroid core structure. We have 4 carbon rings here that are connected to one another, and any molecule that has this sort of a structure within it, something that looks reasonably similar to this, is classified as a steroid. This actual molecule, this is called the core steroid, the steroid, but when people refer to steroids, they very rarely are referring to this specific molecule. They're referring to the derivatives of this molecule here, molecules that have a very similar structure to this one. Okay, right. Now there is some nomenclature that people generally use with regards to steroid structures, and it relates to these 4 rings here. So this first ring here is often known as the A ring of the steroid core structure. This is known as the B ring of the steroid core structure, this one as the C ring, and this one as the D ring. So very simple. And now there's also a numbering system for the carbons of the steroid core structure. So we start off with this carbon of the A ring here. We call that number 1, and then we work our way round. So this one's 2, this one's 3, this one's 4, this one's 5, this one is 6, this one's 7, this one 8, this one 9, this one 10, this one 11, this one 12, this one 13. Oh, and I should have explained how we're jumping here. We are going round. That's reasonably sensible, going round like that and closing the loop. But then, of course, you've closed the loop, so you then go up to here. So this one's 11, this one's 12, 13, and then you go down to here. This one's 14, this one's 15. This one's 16 and this one's 17. So it's a reasonable, whoops, I almost made that into a 12. It's a reasonably sensible path. Starting here, you go round like so to there, then you go on, continue on from here, down like so, and up to there. Okay, so that's the numbering system then for the carbons of a steroid core structure. Right, so that's what a steroid is. Aldosterone then is going to be a steroid hormone. Its structure is going to be highly related to the structure that I've got drawn out in front of you here. So let me now show you how to modify this core steroid structure to turn it then into aldosterone. And by the way, all of these steroid hormones that are synthesized by the different layers of the adrenal cortex, so the aldosterone that is synthesized up in Zona Glomerulosa, the cortisol that is synthesized in Zona Fasciculata, and the sex hormones that are synthesized in the Zona Reticularis, all of them, they originate from the molecule cholesterol. This is what we use to synthesize them. So the cells in these layers use the molecule cholesterol, which is itself a steroid. Specifically, it's a sterol, which is a steroid with an alcohol group coming off carbon-free. So when people say sterol, that just refers to any chemical molecule that has a structure based on the core sterol structure, which is just a steroid with an alcohol group coming off carbon-free here. So sterol and steroid are very similar concepts. Indeed, all sterols are steroids, it's just that not all steroids are sterols. Okay, so cholesterol then is the biologically readily available molecule that we're going to use as the starting point for all of these steroid hormones. Okay, so let me now show you the structure then of aldosterone. So, we're going to have made quite a few modifications here. So I'll start off by talking about what we're going to do to carbon-free here. So we are going to put a double bond to an oxygen atom, a carbonyl group coming off carbon-free here. So at the moment, carbon-free here has 2 bonds to hydrogen atoms in the core steroid molecule. We're going to take those hydrogen atoms off. They're not shown here, but they are there because it's a skeletal structure, we haven't shown them. We're going to take those 2 hydrogen atoms off, and instead we're going to bind the carbon to an oxygen there. So I'm going to start by just drawing out the core steroid structure again, like so. So here's ring A, here's ring B, here is ring C, like so, and here is ring D. Okay, so we're going to start our modifications by putting a carbonyl group onto carbon 3 here. So that's the first modification I'm going to make. Next I'm going to work my way up the molecule like so. Next I'm going to put a double bond in. I'm going to put a double bond in between carbon 4 and carbon 5 here. So at the moment, looking at carbon 4 and carbon 5, we've got 2 hydrogens coming off carbon 4 and we've got 1 hydrogen coming off carbon 5. Take 1 hydrogen off each of them, take 1 off 4, 1 off 5, and instead form a double bond between them, form another bond between carbon 4 and carbon 5 to make that bond there into a double bond. So that's the next modification. In addition, we're going to now go up to this carbon here, carbon 10, and we're going to stick a methyl group of carbon 10. And remember, in skeletal formulae, this is how you show a methyl group. You just need a line to a blunt end, which is going to be the carbon atom there, and that's now going to have 3 bonds to hydrogen atoms. Okay, so carbon 10 would have had a hydrogen initially. We cut that off and put a methyl group attached to there instead, and that's what the next modification is. Working upwards, we're now going to go to carbon 11 here. Carbon 11 would have had 2 hydrogens coming off it. Take 1 of them off and replace it with an alcohol group, slightly more interesting. Okay, then what we're going to do is we're going to go up to carbon 13 here, and we're going to again take the hydrogen off the carbon 13 that would be there in the core steroid structure, and we're going to put a carbon atom that is actually going to be an aldehyde group. So it's going to have a double bond to an oxygen atom, and then it's going to have a hydrogen also here. Okay, and this is one of the reasons this molecule is called aldosterone. It's one of the aldehyde groups that this molecule is actually going to possess. In fact, sorry, no, this is the aldehyde group. Just remembering back to chemistry, I'm thinking these other carbonyl groups count as aldehyde groups, but they don't. This is a ketone group. Okay, this is the aldehyde group, which is the reason that the entire molecule is called aldosterone. Okay, so that's the aldehyde group that underlies the name of this steroid hormone. Finally, off carbon 17 here, we're going to stick a 2-carbon chain here, and I'll just try and fit it in. So here's 1 carbon, here's the 2nd carbon. This first carbon that we put on is going to have yet another carbonyl group, so another ketone group here, and then the 2nd carbon here is going to have an alcohol group coming off it. So this is a 2-carbon side chain here. This carbon's got a carbonyl group, and the 2nd carbon here has an alcohol group. So that then is our modifications complete. That is the aldosterone steroid molecule. So you can clearly see how it's very much so based on this molecule, so we can call it a steroid hormone. Right, just to tell you how to number up the carbons, the additional carbons that we put on here. So the way you do it is we've numbered up all the carbons here. Okay, so the carbons of the rings remain exactly the same. The rings are still intact, so the numbers on the carbon rings remain intact. So we now just need to number up this carbon, this carbon, this carbon, and this carbon, the ones that we've added on. The way you do it is you go to this one firstly, and you call that number 18, so this one becomes number 18. This one here becomes number 19, and then you turn your attention to the side group of 17. So this one now becomes carbon 20, and this one becomes carbon 21. So just highlighting those in. This one to the left of 17 here is then 18, so the one coming off carbon 13. Then we've got 19 over here coming off carbon 10. And then we go finally to the group coming off carbon 17 here, and we call these 20 and 21. Okay, so that's the way you number up the carbons of the additional side chains that are on the aldosterone molecule. So this is the aldosterone molecule. You don't need to memorize it. The main lesson here is that it's based on this structure, and that's the reason it's known as a steroid. In addition, it is synthesized from cholesterol. That's an important point. Now, I did debate whether or not to include the actual biosynthetic pathway from cholesterol to aldosterone, but I have omitted that because it would have taken probably up to an hour. It's not a too long pathway, but it would have taken me a long time to draw all of the molecules out. When you compare it, for instance, to the biosynthesis of cholesterol, it's a very short pathway. It only involves a few enzymes. It is doable, okay? You convert cholesterol, in fact, work out how many steps it involves. You can convert cholesterol firstly into pregnenolone, that's step 1, then pregnenolone into progesterone, step 2, progesterone into 11-deoxycorticosterone, I believe, step 3, 11-deoxycorticosterone into corticosterone, step 4, and then corticosterone into aldosterone. So 5 steps. So it is doable, and if you are interested, do go away, look up the structure of cholesterol, and look up the biosynthetic pathway. It doesn't take too many steps to go from cholesterol to aldosterone. So don't worry about the details of what I've just said. Just understand that it doesn't actually take that many steps to go from a cholesterol molecule to an aldosterone molecule. And neither does it take too many steps to go from a cholesterol molecule to, for instance, a cortisol molecule, which is what the Zona Fasciculata would want to do, or indeed one of the sex hormones, which is what the Zona Reticularis would want to do. Okay, so now then drawing a Zona Glomerulosa cell, so let's say this here, here's its nucleus. This is going to be one of the cells in the Zona Glomerulosa, and it's a classic sort of boring looking cell, a nice circular cell here. Okay, so these cells of the Zona Glomerulosa, they have the angiotensin 2 subtype 1 receptor on their surface as well. So here is the AT1 receptor on the surface of the Zona Glomerulosa cell. And when angiotensin 2 goes up in the bloodstream, these Zona Glomerulosa cells are going to be receiving more stimulation now. So this is going to be angiotensin 2 stimulating this receptor. And that then drives increased synthesis and secretion of aldosterone. So that's an important point. They do not store aldosterone. It's very lipid soluble. Okay, so you can't store it in vesicles in the way that you can, for instance, with adrenaline or something along those lines. So when you get stimulation of the angiotensin 2 subtype 1 receptors by angiotensin 2, it's going to drive the on-the-spot synthesis from cholesterol, and also then secretion. So it drives both synthesis and secretion of aldosterone. So the aldosterone level, therefore, in the blood is going to increase. In contrast, of course, if mean arterial pressure is too high, then we know that angiotensin 2 in the blood will go down, and therefore below its basal level, and therefore the stimulation on the Zona Glomerulosa cells is going to go down, so aldosterone synthesis and secretion is going to go down. So overall, to summarise where we're up to at the moment, if mean arterial pressure is too low, we're going to get aldosterone, this final hormone now of the Renin-Angiotensin-Aldosterone System, going up above its basal level, whereas if mean arterial pressure is too high, we're going to get aldosterone going down below its basal level. Now, what does aldosterone do? Well, aldosterone is going to be involved in raising blood volume. So a raised aldosterone level is going to lead to an increase in blood volume. A lower aldosterone level than normal is going to lead to a drop in blood volume. So this is the one that's going to change blood volume. But to find out how aldosterone actually affects blood volume, you're going to have to wait for the next video. So we'll have a break here and we'll cover the actual mechanism of aldosterone in the next video.